Hi guys, welcome to part 1 of Understanding Cancer. So this video series is going to be about the cell cycle and understanding what goes wrong in the cell cycle and how this can lead to cancer. And so the focus of this video will be on the G1 checkpoint. And the G1 checkpoint comes after the G1 phase, which is the gap 1 phase, which is the phase where the cell will produce its messenger RNA and the proteins it will need to replicate its DNA in the S phase. G1, S, and G2 comprise the interphase, and which is where the cell will spend most of its time. So the G1 phase is of importance because it is the only time where a cell, such as a mature heart cell or nerve cell, can exit the cell cycle and stop replicating. This is also important because, or the checkpoint here is important because it ensures that only cells that are supposed to replicate are the ones replicating, and it also ensures that damaged cells that could lead to cancer are not replicating. So let's go a little bit deeper into the G1 checkpoint. Okay, so when a cell is ready to pass through the G1 checkpoint, a cell signal molecule called the mitogen is released and sent to the cell. So mitogen is short for mitosis generator, and it's basically a signal that tells the cell that the cell has, finished producing the proteins it needs for S phase, so it's ready to pass through G1. And so the mitogen is a general name for a variety of molecules, examples of which are vascular endothelial growth factor and epidermal growth factor. So the molecule that serves as the mitogen depends on what type of cell it is and where it's located in the body, so it's part of the specificity of the body. So this mitogen will bind to a receptor tyrosine kinase, and a receptor tyrosine kinase is again another general name for receptors. They're dimers, so that basically just means that they have these two arm-like structures you see here. And it's another general name. So if your mitogen is vascular endothelial growth factor, then your RTK will be your vascular endothelial growth factor receptor. So, and this receptor, when the mitogen binds to it, it causes a conformational change in the receptor, and this activates the receptor and causes it to activate this protein that is bound to it. And this protein is always bound to the receptor. It's just not activated unless the receptor is activated. So what this RTK will do is it will phosphorylate your GRB2, and that is because RTK is a kinase and kinases phosphorylate. So your GRB2 stands for growth factor receptor bound protein 2, and it is a another protein that is bound to SOS kinase. And it will phosphorylate SOS kinase when activated, and this phosphorylation will activate SOS. And SOS stands for son of sevenless, and it will also phosphorylate its target, which is RAS protein. So we can take a closer look at what happens here. So RAS protein is a G protein, which means when it's inactive, it has GDP bound to it, and when it's active, we can see here that it has GTP bound to it. So GTP has 3 phosphates, as you know, and GDP, guanosine diphosphate, has 2 phosphates. So what the SOS kinase will do is when it is activated through the mitogen's downstream signaling, it will phosphorylate RAS protein. So that means the GDP with 2 phosphates will now have 3 phosphates, and it will turn into GTP. And this means your RAS protein is activated. So this essentially activates the MAPK-ERK pathway, which is a very important pathway in the cell cycle. So the MAP3 kinase here is RAF. It's the first kinase that will be phosphorylated by RAS protein, and it is the third kinase upstream from the MAP kinase ERK, which is what the pathway is named after. So RAS will phosphorylate RAF, activating RAF, which means RAF will now go and it will phosphorylate MAP or MEK1 or MEK2, and this is the MAP2 kinase, and this phosphorylation is an ATP dependent process. ATP will release a phosphate and it will result in the phosphorylation of MEK. MEK, when activated, will release will phosphorylate ERK1 or ERK2, and ERK1 or ERK2 will phosphorylate c-Myc. So it's important to note here that the MAP kinases are held together by a scaffolding protein. So this helps so that the proteins like the MAP3 kinase doesn't have to search around the cell for the MAP2 kinase. It's right there downstream from it. And the scaffolding is also currently being researched, and it's said to help the catalytic function of these proteins through allosteric mechanisms. So that's something that is still new in the field. And so once c-Myc is phosphorylated, it means c-Myc is activated, and c-Myc is a transcription factor. So when it's activated, it will go into the nucleus and it will upregulate the transcription of cyclin D. So cyclins are a family of proteins that regulate the cell cycle. And so basically you can monitor, by monitoring the concentration of the different cyclins, you can kind of figure out what stage in the cell cycle the cell is at. So cyclin D concentration will be really high when the cell is about to go through the G1 checkpoint. So once cyclin D production is upregulated, it will go and it will bind to CDK4 or CDK6. So once this heterodimer complex has formed, it uncovers the active site of the CDK by causing a conformational change in the CDK. So now it's almost activated, but before this complex can be activated, it needs a CAC, which is a CDK activating kinase. And this CAC will come and it will phosphorylate the CDK and cause another conformational change that fully activates the CDK. Once fully activated, the CDK will go and it will phosphorylate PRB, so retinoblastoma protein. And phosphorylation of PRB will cause a conformational change in PRB that will release E2F. And E2F is a transcription factor that will go into the nucleus, and it will transcribe several important targets that will be needed in replication, including cyclin E and A, CDK2, histones, lamins, replication, and repair proteins. It's important to note here that cyclin E is produced. Once cyclin E is produced, it means you have successfully passed through the G1 checkpoint. That cyclin E is involved in a positive feedback loop. So what happens is cyclin E will bind to CDK2, and when it does that, so let's see, CDK2, CDK2. And this complex, once activated by a CAC of course, will go and it will also phosphorylate PRB again. And so you have more E2F being produced and you have more cyclin E being produced, and this results in an amplification of the response. And now the checkpoint has been passed. So the next step is to look at how things can go wrong at this checkpoint or within the checkpoint, and how that will lead to cancer. So this concludes the first video.